Good morning, everyone. We're broadcasting live from the BD2K Hackathon in San Diego.
Yeah, so instead of starting a little later, we'll start now. So good morning, everyone, for those in the US, and good afternoon to those in the UK. Today we're going to have two presentations, one about the details of the second round, and the other about collaborations with ProTurn, and that'll be more of a discussion. We are scheduled to start at 8 a.m. Pacific time and end at 9 a.m. Pacific time. And these conferences will be recorded and accessible online. And if you have any questions during the presentation, please type it in the message box provided in the app. Also, please mute your mic during the presentation to prevent any background noises.
Just try to switch my screen to yours.
Yeah. Okay. So last week, Brian and I presented a few of our ideas on the focus of our second round. We received a lot of good feedback about our ideas, and we've come up with a structure that will focus on collaborations. So I have outlined here the four goals that we set for these technical conferences. So in the first round, we focused on learning about the existing tools. And for our second round, we'll focus on the next goals, prioritizing promoting collaborations among tools. And initially, we proposed on focusing on more technical-based presentations, but because we all have the basic knowledge of each tool, we can start thinking about collaborations and hopefully have new projects going. And then maybe we can have presentations about updates on these collaborations to keep us all in the loop, and possibly more technical presentations to keep us to help us with these projects later on. And here we have the conference structure for the second round. So most of it is the same, except we have discussions instead of presentations and a Q&A session. So for each conference, we'll have two discussions, one for each tool. And these discussions will be about 30 minutes each. If we run out of time, these discussions can be carried over to the open questions or discussion sections starting at 7:30 to 8 or 9 to 10. So in each discussion, there will be three parts. First, the presenter will give a brief overview of the tool, and most people should be fairly familiar with the tool at this point. But if people would like more information about the tool, they can access the first round presentations on the YouTube channel. And I'll also provide a link to the videos in our weekly updates. The presenter will also quickly go over some of the current collaborations that he's involved in. And lastly, the presenter will be leading a discussion for possible collaborations with other tools, and I'll go over that in just a bit. So for the discussion, we have three different roles. We have the presenter, who is presenting the tool, giving a quick overview, and also proposing a few ideas for collaboration if he has any. Next, we have the moderator. And although a moderator may not be necessary for some of these discussions, but the basic idea of a moderator is that he would be someone who is an expert or knowledgeable of the field that the tool operates in, or even just a primary user of the tool. And the moderator would be facilitating these discussions and comment on the proposed collaborations. Again, I'm not sure if a moderator is needed, but I think it will be helpful to have someone providing feedback for the collaboration. And if the discussion doesn't have a moderator, then the presenter would also act as the moderator for the discussion.
So I have a question. So how would we assign these moderators?
So the presenter would choose the moderator. So
Oh,
he
okay.
can ask like a like a like a user of that tool, and be present so
Okay,
that
that's a good idea.
they can give feedback. And last, we have the tool representatives who are looking to discuss about the potential collaborations, and all attendees of the conference would be considered tool representatives, and they would be participating in the discussions with the presenter, and hopefully form potential collaborations. So in the weeks leading up to the discussion, a presenter would have a few items to prepare. He would create a few slides for a brief overview of the tool, and also mention any collaborations that the tool is currently involved in. And the presenter would choose one or more tools that he thinks could form a collaboration and propose ideas for how tools could work together. So this would lead to discussion among the presenter, moderator, and tool representative. And the presenter could also choose a few tools that he thinks he could collaborate with, but not necessarily have proposed ideas for. And this is generally to spark discussions for potential collaborations. And lastly, the presenter would also choose a moderator if she chooses to have one. So here I have a simple animation of a discussion. So first, the presenter would propose one or more collaboration ideas, and he would have a discussion. He would be having a discussion with the tool representative, as well as the moderator and anyone who would like to contribute their ideas. And if we don't have and if we have time left, the presenter would discuss with other tool representatives that could potentially collaborate with the tool, and this would generally be a very an unstructured discussion, but the presenter would just hopefully lead the discussion and talk to some of the tool representatives to come up with new ideas. And the goal of these discussions is to form hopefully at least one collaboration, or at least to begin discussions for potential collaborations. And here I've outlined the schedule for the presenter. So about 2 weeks before the presentation day, you'll receive a reminder, and the presenter would also send the list of potential collaborators so that I can email them and make sure that they will be present at the conference. If they are unable to attend, then we could set up a time during the open discussion portions of our conferences to hold a discussion for the collaboration. That's about it. Thank you for listening. It would be great to hear your thoughts and feedback for the new conference structure, or if you have any questions, I'd be happy to answer them. Okay, is everyone okay with this conference structure? I guess no feedback. Okay, well, we'll have Brian present about ProTurn and its collaborations. So you guys will see, a demonstration of how the new conferences should be.
Hi guys. So I'm going to be talking about ProTurn and our potential collaborations. Just going to, like I said, give a brief overview of ProTurn for those of you who missed the first presentation or don't remember. So ProTurn's a tool that measures protein turnover rates for in humans and mice. So the organism you can see in the top left here is enriched with heavy water over a period of days, and blood samples are taken at the different time points, and they're analyzed by a mass spectrometer. And as you can see, further down the experiment in later days, you see the peaks start to shift to the right as more and more heavy water is incorporated into the organism's proteins. And then we measure those peaks and come up with a ratio of these higher order peaks to the initial peak, and that ratio is going to increase as more and more heavy water is absorbed by the organism. So then we can take proteins and we can calculate the turnover rate for individual proteins in a sample. So obviously the inputs for ProTurn, we need so MZML files for an experiment, and also we need search engine results. And we need a series of those. Well, actually, it can actually function with just a single time point. So if you've been, so like if you've been giving an organism heavy water for 7 days, and on the 7th day you take a sample, we can still use our model to calculate the turnover rates, because we obviously know just from the periodic table, we know the initial relative abundances of all different isotopes for, carbon, oxygen, etc. Okay, so our current collaboration. So right now with COPA-KB, ProTurn can currently download and analyze COPA-KB data. So as you know, COPA-KB has a library search online. People can upload MZML files. You can use ProTurn to download data directly from COPA-KB with your task ID. So that functionality currently exists, but we're in the process of overhauling the COPA-KB API. So we'll update ProTurn as we go, because both products come from the same laboratory. So it should be easy to keep those in sync as they develop. Okay, the next most obvious collaboration is with IP2, the Integrated Proteomics Pipeline. I'm not sure, is anybody in the audience here with IP2? Probably not. I think most Scripps people are here at the hackathon.
Yeah.
So I guess not. Well, so IP2 is a commercial enterprise. Users upload large numbers of MZML files, and it runs ProLucid, DK Select. I think Census is integrated, and it gives you comprehensive search results for your raw data. So they have a development kit which lets users make a plugin. So this is probably the most straightforward collaboration we could do. It would be nice if users could upload their data, and as soon as the search engine results are done, they could start quantifying their turnover rates with ProTurn if they uploaded a whole series. That would be helpful, definitely. Okay, the next most obvious collaboration, I think, is with Pride and Proteome Exchange. So obviously that's a repository for both processed and unprocessed data. So Pride and Proteome Exchange, currently they accept data linked to publications. And generally, ProTurn's kind of analysis you would do on your data before a publication, because it's for a specific experimental setup. So in that sense, the collaboration is a little less straightforward. But as part of the B2K grant, there's already a planned reverse expression interface. And I got the description from the grant right here. The idea is that you upload an expression data set in the same format as the expression interface, and as expression interface return format, and you upload it to another resource for analysis. And then the proposed resources in the reverse expression interface include, a Reactome, ProTurn, also IP2, if IP2 was connected with this reverse expression interface, and then we had a ProTurn plugin, then that'd be easy because you could go from Pride to IP2 to ProTurn. Because ProTurn is dealing with search engine results. Those are the three, the three most direct collaborations I can see, because IP2 and CodeBookKB both search raw data and give results to the user, and Pride and ProTurn Exchange are a repository of both the raw data and the search engine results. Those are the most straightforward collaborations I see. If you really want to have sufficient input to run ProTurn, you need both your search results, but you also need your MZML files, because you have to go in and look at the original spectrum to actually look at the peaks to see how the peaks are shifting between each time point. So, does anybody have any idea, how we could connect ProTurn to any of the other tools in our center?
Hi guys, this is Sarah. I'm not certain I have insight for you regarding how, but I can tell you that I think a really useful connection would be between ProTurn and BioGPS. From a user standpoint, I think being able to visualize all the different analyses in one window for comparison purposes would be really nice. Have you thought about that at all?
So just so basically the ProTurn results would be exported into BioGPS for visualization.
Exactly. Yeah, that's what I'm asking. Is that doable? High impact to labor ratio.
Yeah, that's definitely doable. Yeah, we have
You can
some
think about it.
basic, like,
Okay.
some graphs
Go
and things
ahead.
like that, some basic plotting, like in the ProTurn application. But
Uh-huh.
yeah, definitely we could try to uh, try to connect with BioGPS to get like a more sophisticated visualization. That's a good idea.
Yeah. I think as we develop these tools and we enhance the communication among the tools, again, from a user perspective, rather than being overwhelmed by all the different tools, if you have a way to graphically visualize all of your analyses in one window, I think BioGPS is a great venue for doing that. I don't know, what are your thoughts on that?
Hmm.
So I guess for ProTurn, you guys would have to create a web interface first, and then from there, um, create a plugin in BioGPS.
Hmm.
I
Yeah,
guess
that's right.
this
Currently,
brings up another.
ProTurn's offline. Uh, it just runs on a user's local machine. Um,
Oh, okay,
but we have been considering ideas of a web interface. Um,
okay.
and
Because
definitely the more the more tools that we connect ProTurn with, and the more we integrate it with the other tools in the center, obviously the more reason there is to have a web interface. It certainly makes all those connections easier.
okay, yeah, just a thought.
Hmm.
What's the I know this was brought up in the initial round of technical talks. Um, the paper's published. Um, how how well has ProTurn been adopted by the community? I think at that point there wasn't really there. You had some interest, but there weren't really too many other users other than our lab of ProTurn. Is that correct?
Yeah, that's correct. We've been mentioned in a few other publications, but I know I don't think another laboratory has actually um, started an experiment with ProTurn yet.
Okay. I didn't realize that I guess it makes sense because you're just, based on the fitting of the curve, but I thought there were a minimum of 3 labeled time points in order to ProTurn. I guess
Um,
anything is going to increase with your accuracy will increase with your number of time points, but
yeah, so there's no set minimum. So obviously, yeah, more time points are better, but
uh-huh,
so at like I don't know if you can see my mouse cursor right here. But yeah, so at time zero,
uh-huh.
there's a theoretical M0 to MI
Okay,
ratio, right?
okay,
Because you know the chemical composition
right.
of a peptide, so you can just based on natural abundances of different isotopes, you can calculate what the M0 to MI would be.
Right,
So most experiments that we've done, we do take a actual um, day zero sample too, uh,
right,
which generally matches pretty closely, though not exactly to the theoretical time point. So
right.
because of that,
You can
and
use
actually if you're doing a certain type of tissue, like not a blood sample, but actual cardiac tissue removed from
it.
during a heart transplant or something like that, you can actually still use the protein analyses even if you only can be able to get one sample.
One
Um,
sample. Yeah. I thought, and I understand that, I thought that the software itself had put that restriction in, but I'm incorrect. I don't know where, maybe that was in when we were doing the initial development, like a few years back. We were sitting in lab meeting, we were talking about that. Maybe not,
yeah,
though.
I mean that is there's a hmm.
I mean, I guess it's like use at your own risk. Like you need to understand that with more time points that your data will be more accurate, right? But it's kind of like,
Yeah, I think there's like that's a setting. There's like because we use like this Apache Math Commons for like um the function optimizer to like set whatever the k value is, and I think that like the number of time points is like a restriction in like that math library,
okay.
but we by adding like the virtual time point, I think it works around. I'm not sure, but we have done like tests with the single time point. But I think that might be in an earlier uh, like stage of development. I think you're right.
Okay. Yeah, this is great. I mean, I'm trying to think, I think as a lab now, it's just, helping the community to understand the value of protein turnover. It's sort of this missing dimension in proteomics that tells us things that expression can't tell us. So I think, I mean, I guess another question, does somebody else want to talk? I can be quiet. Speak now. um, okay, I know we have a limited number of attendees today, so
Yeah.
I guess what's sort of the plan for raising enthusiasm in the scientific community for using this tool?
Hmm. Um. Well, I mean, I guess it's just I mean, I don't know the plan myself. I think it's just a combination, of the our publications getting more exposure, and also I think once we're able to integrate it with other tools of the center, uh, it'll sort of be able to get more exposure through that avenue. Um,
That's right. I agree with that.
hmm,
Okay, cool. Thanks, guys.
yeah. Thank you for your input.
Yeah.
So I mean that's a little bit brief. I guess that's sort of an example of the kind of collaboration presentations. Yeah, obviously because of the hackathon, a lot of people aren't here. and that'd be helpful to get input from from IP2 and everything.
Yeah. So ideally we'd invite the mentioned collaborators that you talked about. So Pride, IP2. So yeah, we would invite those people, and then hopefully we would have a discussion. So we can hopefully carry that discussion on next week because they're not here. Can't really discuss it further, but I'm sure those are great ideas. So I think it'll be better if we talk to them about it. Hopefully next week. But does anyone have any other questions? So this is a basic demonstration of what it'd be like. Obviously with more discussion, but due to the small attendance, couldn't really do that. But I'd like to. Before we end, I'd just like to show you just the presentation sign-up form. It's been updated for this second round. All right. So we have just a simple form. Put your name, email, put in your institution, put in your tool, and also a short description of the tool. And here we have so here you can input your current collaborations that you're currently involved in within our PB2K Center. And if you'd like, you input a moderator for discussion, and I think the moderator would be really helpful in giving feedback, especially on the user perspective. So if you can try to invite someone to moderate the meeting for you, so that we can get more user feedback. And then last we have the preferred discussion day, and this is where you select four days or more, and then after that I'll look at all the signups and hopefully create a schedule based on that. Are there any questions? Okay, thank you all for coming. I really appreciate that you guys are taking the time to come and attend these conferences. Hopefully next week we'll have more people and we'll have more discussions about these collaborations. But today was pretty good. Sarah provided a lot of good feedback, proposed a new collaboration, and I think that was already pretty productive. So thank you all for coming.
Yep. Thank you, everyone.